Hello, my name is Christian Coviello. I'm the CTO of Oxonics, and I'll be talking about the validation of our product, SonoTran. The problem that we're trying to address is that solid tumors are poorly penetrated by oncology agents, owing to a tumor's poor and irregular blood supply and high interstitial fluid pressure. Our solution, SonoTran, combines a formulation of cavitation nuclei plus focused ultrasound to create a targeted micropump that can significantly enhance penetration of oncology agents into solid tumors. Across a range of cell lines and a range of therapeutics, we've seen enhanced tumor uptake leading to increased survival and tumor size control. Safety of our system has been demonstrated in a GLP tox study, and as well as some large animal experiments I'll show the results of later. Part one of our product, our SonoTran particles, are shown here in the TEM image. Each particle is 450 nanometer in diameter, cup-shaped, having a 200 nanometer cavity. Each of those cavities stabilize an individual air bubble. Those air bubbles, when excited by focused ultrasound, expand and inertially collapse. We don't see stable cavitation from the particles. The inertial collapses lead to microjetting, pushing both drug and particles away from the site of inertial cavitation. How does it work in practice? Well, a patient arriving at an oncology ward would receive into one arm an infusion of our particles, into the other arm whatever it is the drug that they're prescribed for their particular cancer. It's completely drug agnostic and there's no reformulation. In theory, this could work for any type of drug. The particles and the drug mix in the bloodstream and make their way to the solid tumor. We then use our ultrasound system to image where we want to apply focused ultrasound, deliver a therapeutic burst of ultrasound, causing inertial cavitation from our particles, pushing both drug and particles deep into the tumor. How do we achieve this? Well, the second part of our system is shown here, consisting of a cart-based unit, as well as a novel handheld probe that has three functions. One, diagnostic B-mode imaging. Two, longer duration focused ultrasound therapeutic burst that can cause cavitation from our SonoTran particles. And then we can actually listen in real time using an algorithm that we've developed called passive acoustic mapping to spatially monitor where that is occurring, as shown by the color map overlaid on B-mode in this image. On the right, you see our software, which we've worked on with our interventional radiologist consultants to ensure that it's fit for purpose and is easy to use. And we've developed enhanced therapy guidance, live registering ultrasound to pre-collected CT or MR. We validated this, that we can cause cavitation and that cavitation is safe in large animal models. So 8 pigs have had their livers and pancreases treated, an example of which is shown here on the right. In the bottom video, we're delivering focused ultrasound into a pig's liver, but you don't see any evidence of our passive acoustic mapping showing that cavitation is occurring because we haven't started to infuse our particles. When we do infuse our particles, immediately we see a color map displayed at the crosshair showing the location of focused ultrasound, showing that we're causing cavitation in that region. We've been able to demonstrate that cavitation can be sustained using our system for treatment times of up to an hour. But most importantly, this doesn't correlate with any adverse events assessed through blood biochemistry, macroscopic assessment of the treated tissue, and post-treatment histology. Additionally, we've been able to validate in Neuron Models. We have comparable data sets across a range of therapeutics and a range of cell lines. This particular example is delivery of an antibody cetuximab that's important to us, where we've shown between a 3 and 4x increase in tumor dose corresponds to an increase in median survival, as well as improved size control and even some cures in the SonoTran treated group compared to control. So in summary, SonoTran can enhance the delivery of unmodified and reformulated drugs into solid tumors using a purely mechanical mode of action. The safety of and instigation of cavitation has been demonstrated in pig models, where we've shown that cavitation can be sustained for up to an hour. We've shown in murine models across a variety of cell lines. Particularly, I showed an example of cetuximab leading to increased survival, tumor size control. Cetuximab is important because we will enter our first in human clinical trial treating metastatic colorectal cancer using cetuximab in early 2021. Thank you very much for listening.